Today we will discuss something about the immunoglobins. So there are our immune system depends upon two types of cells, B lymphocytes and T lymphocytes. B lymphocytes are formed in bone marrow cells in higher animals and from the bursa of Fabricius in birds, and immunoglobins are synthesized in B lymphocytes. T lymphocytes, these are of thymic origin and these provides the cell-mediated immunity, whereas the B lymphocytes provide humoral immunity. This is the basic structure of immunoglobin. This is the immunoglobin G. There are two light chains and two heavy chains, and there is some sulfide linkage, and these portions are joined by the hinge regions, and enzymes like papain and pepsin can digest and can give two breaks in this one. One portion is called as the antigen binding regions, and other is the crystallized FC regions. Light chain is the product of at least three separate structural genes: a variable region genes, a joining region genes, heavy chain at least four different genes: variable region gene, diversity region gene, joining region gene, and constant region gene. The L chain and H chain are synthesized as separate molecules and are subsequently assembled within the B cells or plasma cells into mature immunoglobulin molecules. The immunoglobulin G represents the typical immunoglobulin. The molecule consists of two light chains and two heavy chains. Each light chain consists of a variable and a constant region. Heavy chain also have a variable region and a constant region, which is divisible into three domains: CH1, CH2 and CH3. CH2 domain bears the complement binding site, whereas the CH3 domain possesses site attaching to the receptor on the neutrophils and macrophages. The antigen binding site FAB is formed by the hypervariable regions of both the light and heavy chains, which are located in the variable regions of these chains. The light and heavy chains are linked by disulfide bonds, and heavy chains are also linked to each other by the disulfide bonds. All immunoglobulins contains a minimum of two light chains and two heavy chains. The half of the light chains towards the carboxyl terminal is referred as the constant region, while the amino terminal half is variable region of the light chain. The digestion of immunoglobulin by the enzyme papain produces two antigen binding fragments FAB and one crystallized fragment, which is responsible for the function of immunoglobulins other than direct binding of the antigens. Because there are two FAB regions, immunoglobulin G molecules bind two molecules of antigen and are termed divalent. The site of the antigen to which an antibody binds is termed as the antigenic determinants or epitope. The five types of heavy chain determinants immunoglobulin classes are immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin G, and immunoglobulin E. Immunoglobulin G represents the typical class of immunoglobulin. It crosses the placenta, main antibody in the secondary response. It opsonizes bacteria, fixed complement which enhance bacterial cleaning. It neutralizes bacterial toxins and viruses. Immunoglobulin A, it is secretory. Immunoglobulin prevents attachment of the bacteria and viruses to mucous membrane. It does not fix complement. Immunoglobulin M, produced in the primary response to an antigen. Fixed complement, does not cross the placenta. Antigen receptor on the surface of B cells. Immunoglobulin D, it is uncertain. It is found on the surface of many B cells as well as in the serum. Immunoglobulin E, It mediates immediate hypersensitivity by the release of mediators from the mast cells and the basophil cells upon exposure to antigen. Uh, Immunoglobulin E defends against worm infection, does not fix complement, and the main host defense against the helminthing infection. So, immunoglobulin M and immunoglobulin G can fix complements. 5 classes of heavy chain have been found in human, distinguished by the differences in their constant heavy regions. They are designated as the gamma, alpha, micro, delta, and epsilon. So this is the question of December 15. There are 2 columns, first and second, and we have to match the immunoglobulin with their respective properties. A binds to macrophages by the Fc. So macrophages binding is most probably the immunoglobulin M. Binds to mast cell and basophils, immunoglobulin E, that should be the third one. No major specific function known other than the binding, other than antigen binding, is immunoglobulin D, and protect mucous membrane, immunoglobulin A. So answer should be the fourth one. Okay, so there are 2 terms in this discussion. There is FAB, that is the antigen binding fragments, and that is FC, that is the crystallized fragment. These are produced when papain and other enzymes releases the J region, joint region, okay, into 2 fragments. So thank you and good luck.